After fertilization, the zygote undergoes cleavage. The sea urchin egg, like all eggs, contain an animal-vegetal axis. However, the animal pole and the vegetal pole are not readily discernible, except by the fact that the egg nucleus or zygote nucleus resides near the animal pole. In the sea urchin, the entire egg undergoes cleavage, or also known as holoblastic cleavage. The first cleavage plane passes through the animal-vegetal axis and splits the zygote into two blastomeres. The second cleavage plane also passes through the animal-vegetal axis, but is oriented perpendicular to the first. The third cleavage plane produces 8 cells and is equatorial. Experiments reveal that individual blastomeres are no longer equivalent at the 8-cell stage, and that isolated blastomeres can no longer form an entire larva, only parts of the larva. Cleavage becomes irregular after the third division occurs. Thus, in some species, there is a distinct 12-cell stage preceding the 16-cell stage. When the fourth cleavage is fully completed, 3 tiers for a total of 16 cells are established. At the tier nearest the animal pole consisting of 8 blastomeres that are now called the mesomeres. At the tier nearest the vegetal pole consisting of 4 smaller blastomeres that are now called the micromeres, and a tier in between consisting of 4 larger blastomeres that are now called the macromeres. Cleavage ultimately establishes 5 tiers of cells for a total of 32 cells. The 8 mesomeres in the animal hemisphere divide to form 2 tiers of 8 cells each. The upper tier of 8 cells are now called the AN sub-1 blastomeres, and the lower tier of 8 cells are now called the AN sub-2 blastomeres. The 4 macromeres divide longitudinally to form 1 tier of 8 blastomeres, all of which are still called macromeres. The 4 micromeres divide to form 2 tiers of 4 cells each. The upper tier of cells are called the large macromeres, and the lower tier of cells nearest the vegetal pole are called the small micromeres. The sea urchin embryo between 32 and 64 cell stage can be called a morula, that is, a ball of blastomeres. The sixth cleavage ultimately produces 60 cells, not as the expected 64, because the 4 small micromeres, unlike all other blastomeres of the 32 cell stage embryo, do not divide during this cleavage cycle. During subsequent cleavages, blastomeres become arranged around the central cavity called the blastocele, and the developing embryo at this stage is called the blastula. That is, with formation of the blastocele and blastula, the process of blastulation occurs. Cilia develop on the outer surface of the blastula. Those at the animal pole become particularly long and are called the apical tuft. In the late blastula stage, cells begin to detach from the vegetal pole of the blastula and to move into the blastocele. That is, these cells undergo ingression. The ingressing cells are called the primary mesenchyme cells. Approximately 64 primary mesenchyme cells eventually form. During the late blastula stage, when primary mesenchyme cells are ingressing into the blastocele, the blastula is often referred to as mesenchyme blastula. The blastula begins to rotate within the fertilization membrane at the late blastula stage through the action of its cilia, which beat synchronously. The blastula produces an enzyme at the baptist stem called the hatching enzyme. The hatching enzyme weakens the fertilization envelope, and movements of the blastula eventually rupture the fertilization envelope, allowing the ciliated blastula to hatch.